Hello, everyone. My name is Jeffrey Blay. I'm a second year MESC from Professor Sito's lab and a member of BIOMES committee as well. I welcome you all to the first seminar of the for the 2023 BIOMES seminar series. A couple of announcements before I introduce our guest speaker for today. First, if you are joining us from Zoom, you can kindly post your questions and suggestions in the chat board and we'll address them. And then also, there are 4 slots available for our lunch meeting with our guest speaker for today, which is slated on Thursday, next week Thursday, 12 noon. So if you're interested, you can sign up for it. And then last but not the least, the BIOMES committee is looking for new Master's NME PhD student for the fall 2023. So if you're interested to be a member of the BIOMES committee, you can kindly reach out to any of the BIOMES committee members, including myself, and then work with us. Talking of BIOMES, our primary goal is to just inform conversations within the YSE community, mainly focusing on community sourced and then interdisciplinary research, interdisciplinary presentations on research policy and then personal environmental issues. Now to our guest speaker for today. He has worked on, our guest speaker for today has worked on carbon in rivers since his undergraduate thesis on the Hudson River in the early 1990s. His research included some of the very first direct measurement of carbon dioxide in rivers. He received a PhD in oceanography in 1999 from William and Mary, studying the carbon cycle of the York River. He has been a professor here at YSE for the last 20 years, where he has continued to study rivers around the world and interact with students around the physical sciences. Fellow YSEs, let's welcome Peter Raymond.
Thanks, Jeffrey, and thanks everyone for coming. So what will do? I have to just reshare the screen, or? Okay. Perfect. So I'm going to talk about rivers and carbon. For today's talk, I have a little bit of like a broader introduction that I'm going to give to sort of orient folks to what I do in my lab. And then I'm going to like funnel down for the second half of the talk to sort of get into a thread of my research in a little bit more detail. But I'd like to start by talking about watersheds and drainage network systems, because it does link into the second half of my talk, and I think it's also a fairly fascinating area. For those of you who've been in my office, you've seen this up on my wall. This is the US as viewed of the drainage networks that make up the US. And they come in all sorts of different shapes and sizes. But one of the things that has come out of this is watersheds and drainage systems have basically developed to have a single form and proceed of a single function, right? And that is to like efficiently move rainwater off the landscape. And that is why maybe the term drainage network is more apt than watershed. They've formed through erosion, through centuries and millennia, to optimize and efficiently move water off the landscape. And as we'll see, that's similar for other systems that have optimized to move fluids around. But one of the things that is important that comes out of this is they have a fractal, self-similar geometry. That's a little bit hard to get into in slide 3. But what that means is if you zoom in on a watershed, so if you have a really large watershed like the Mississippi, if you zoom in on a piece, the geometry of that watershed is the same as you zoom out. So the physical components of that watershed are self-similar and fractal, or another word that people use for this is they scale, right? And as you're learning here, as scientists, one of the really important things that we try to do is scale our science. And that is one of the great things about working on river systems and drainage networks, is they have this scaling inherently built into them in their physical structure. And I'm going to just sort of walk through that a little bit more to make sure it's clear with folks. Like most things, this was first touched upon by Da Vinci, right? A great scholar, touched upon a lot of different things, and he recognized both the self-similar nature of watersheds and also the similarity between different systems that have evolved to move fluids around. He called water systems veins of water. But notice that they were similar to the circulatory system of the body, right, which has evolved to move blood to our extremities, the veins of the arm, and also the branching patterns of trees, which have evolved to move water from the roots to the atmosphere. If you've read this, he had language that was sort of touching upon the fractal mathematics of drainage systems. More recently, this was touched upon by Maya Lin. She had a really cool exhibit in the Hudson River Art Museum, where she was also really focusing in on sort of the form of watersheds or drainage networks or the patterns that evolve. She named her exhibit The Folding of the Hudson, and had a few pieces that were sort of, really focusing in on these drainage patterns and these branching patterns of the river and the watershed, and these were done in marbles over a room. But for us science geeks, like, this was really sort of laid out in the early to mid 20th centuries. There were a few scientists who were like really focusing on the geometry of watersheds, the geometry of streams, and a lot of credit is given to Strahler, who sort of codified this in the late 50s. And there were a few things, there were a bunch of things that came out of this paper and built upon other works. One is the ordering system that develops when you have these sort of self-similar branching patterns, and what we call scaling laws. And I'm just going to walk through these a little bit, but you can see, I'm not going to go over this in detail, but you can see Strahler in this paper working out the self-similar geometry of watersheds, where the small watershed on a plain has similar geometry to the larger watershed that it's embedded in. But many of you are probably familiar with the branching system. The same branching system occurs in literature of the circulatory system and of tree branching systems. And the way it works in streams and rivers is the very first streams that emerge off of the landscape, where you have surface flow, we call stream order 1, right? So here you are here, our headwater streams. Like I said, the smallest streams on the landscape. When two first order streams meet, you form a second order, right? So you can see that here. And then when two second orders meet, you have a third order, and so on, up until the Amazon, which is a 12th order stream, depending upon who you ask. You can have first orders connected directly into third, second orders connected directly into eighth. But what happens is, due to this self-similar nature, these scaling laws emerge. And there are a number of scaling laws. I've listed just 3 here, 3 ratio scaling laws. And the way these work is, let's just go over the ratio scaling law for length. That is, if you were studying a larger watershed and you wanted to know what the average lengths of your different stream orders are, if you just went and, say, measured 10 stream orders 2 and 10 stream orders 3, you concluded that these were on average 2 kilometers and these were on average 4 kilometers, right? You'd have a stream length ratio of 2. Without going and measuring any other streams, you would be able to conclude that the length of your 4th orders would be 4, the length of your 8th orders would be 8, and so on and so on. And that's due to the way these systems have evolved and the self-similar geometry of the systems. There are also scaling laws for the area of those watersheds, right? The contributing area, the number of those stream orders within a watershed, even things like velocity of the streams, depth, and so on. And as you'll see, we're going to leverage that fairly heavily in some of our research where we're trying to conceptualize even biogeochemistry. I just wanted to also step back and like think a little bit like if you're studying a drainage network, the variability that you might see. This is a really cool photo that a master's student, Elizabeth Creech, made in my lab maybe 5 or 6 years ago with a fisheye lens. This is what we use to try to figure out how much light is hitting the stream. And you can see she's standing in a first order stream. If I hadn't told you that, right, you wouldn't know it. Like you're basically in a forest. And so when you're down in the little streams, you're really studying in part the biogeochemistry of the forest or the agricultural system, and the things that are going on in that watershed are really important to the chemistry of that stream. And you could take it all the way to its extreme and look at the Amazon River, where if you're studying the Amazon River, if you're an oceanographer, right, in the North Atlantic, you need to know something about the Amazon due to the amount of water that it's putting out into the coastal ocean, right? And so I'm trained as an oceanographer, and there's a subset of oceanographers who study rivers because they're this really important sort of input term. But obviously the size of the system that you're dealing with is, almost like a completely different system than that small headwater stream. Okay, so I just wanted to go over a few different reasons what motivates me in some of my research over the years to study carbon in rivers. One is, like I said, they are a major input to lakes, reservoirs in the coastal ocean. This is a really cool photograph of the Connecticut River after Hurricane Irene, showing the sediment plume, right? It only hit the western part of the Connecticut River watershed, right? Didn't hit the Thames watershed at all. You can see this had impacts that lasted for many weeks to even years. So it's important if you are studying systems like that receive river water to understand the chemistry of the river water. It's also like there's an analogy often used that studying the chemistry of rivers is similar to doing blood work. And similar to doing blood work on ourselves, it provides sort of like an integrative view of what's happening to our body or the watershed. And there are a few different things you can gain at that. One is if you're really lucky, you can have a time series. And over time, if your chemistry starts to change, it's an indication that something in your system is starting to change for good or for bad. And this is an example from my research where we noticed that the concentration of bicarbonate ion in the Mississippi River was changing, due in a large part due to agricultural activities in the watershed. But you could also say, go out and survey a bunch of patients' watersheds and look for outliers. And that might give you an indication that there's something different in that watershed that you have to then go out and go into and try to tease apart. But it's also important to remember that these are really interesting and cool ecosystems in and of themselves. They have a sort of group of organisms that you obviously don't see anywhere else. And carbon is important to a lot of different sort of processes within streams and rivers, and also even important to sort of like the physical component of the system, things like light penetration. How much light penetrates in a water system is partly dependent upon the amount of carbon in it. And so I'm not going to linger a lot, but some of the questions we've been looking at in my lab over the years is I sort of started off in part, like I said, looking at CO2 coming out of streams and rivers. I've also looked a lot at how old carbon is in rivers. But then some of the fundamental questions of how is managing land changing river chemistry, how much is making it out to the ocean, what controls that variability, and how does either carbon concentrations impact river processes, or how can we learn about river processes by studying carbon chemistry. Most of these photos are from river systems in the Connecticut River. This is a cool one, right? Because it shows a US gauging station. For those of you who are familiar, these are the things that the USGS have out all around all our watersheds in the US that measure stream discharge. They're not that pretty usually, but it is what. And so what I want to do now then is just focus down, and like I said, into one thread of my research over like the last 10 years. And it's focusing in on river organic carbon. And to remind everyone, organic carbon is basically organic carbon that's produced by primary producers. It contains, it's reduced, it contains energy within it that can be focused in on by biological organisms as an energy source, right? In a river, we sort of divide it up a lot of different ways. Two general ways we divide it up is sort of by size. So you'll go out and you'll talk about and you'll measure particulate organic carbon. So this is a particle in the water. So it has the chance to be deposited and resuspended. And so has a lot of different sort of transport characteristics than the other type, which is dissolved organic carbon, which is organic carbon that's dissolved in water. This is maybe a little harder to think about, right? So I have this thing on the right. It's often called, dissolved organic carbon is also called watershed T. So you can think about it like as soon as rainfall hits a watershed, it starts dissolving carbon from the leaves of trees and then from the soils, and gives this water often this characteristic brown tinge that you see. The other way that we often divide it, and this is going to come in a little bit towards the end of the talk, is by its source. And there are two main sources of organic carbon in rivers. One is watershed T, so terrestrial organic carbon. The other one is organic carbon that the plants within the river itself produce, right? So things like submerged aquatic vegetation or phytoplankton are also organic and producing particulate and dissolved organic carbon. DOC in particular, and this gives you like two great photographs, right? These are two Connecticut watersheds. You can see the brown color of the streams and rivers. That's mostly dissolved organic carbon, giving it that brown color. Also iron provides some color to water. But it provides a lot of different, it's important for a lot of different reasons. Like I said, you can obviously see it's going to absorb light. It also absorbs UV, which is harmful to insects, just like it's harmful to us. It has a lot of heavy metals associated with it because it has a lot of binding sites. So like the transport of mercury is in part determined by the transport of dissolved organic carbon. Bacteria utilize this as their food source. It also has a lot of organic nitrogen and organic phosphorus associated with it. One of the interesting things also is that in a lot of water treatment processes, the DOC basically forms a lot cancerous byproducts. And so it's focused on and regulated in your drinking water. So I wanted to start with a DOC study that Jim Sayers and I did in 2010. This was like my first metadata analysis, right? And back then, a metadata analysis with 5,000 measurements was a lot. It's not necessarily a lot today. And what we did for this study is we sort of asked a simple question, is we went into all the USGS sites that were almost all forested, downloaded all the DOC data, matched it up with the amount of water coming out the watershed, and we're just asking simple questions of, what is a characteristic forest? How does its DOC export? What does it look like? And what we found was, and others had found this before us, right? But the power of the metadata analysis is you begin to show that it's something that's common or general amongst systems. What we found was that as you had storms, which makes discharge go up, right? So if you're in a stream, you get more and more discharge as you get a larger and larger precipitation event. The concentration of DOC responds by also going up. And this happens with some other elements, but it's fairly abnormal, right? And so what this means with respect to the movement of DOC off of terrestrial landscapes is these storms become very important, right? Because not only is there a lot of water coming off, but the concentration is going up when a lot of water is coming out. And so because this were USGS sites, we were able to sort of actually quantify this, and we concluded that in just 15 days of the year, 60% of your DOC is coming out of these headwaters and out of these terrestrial watersheds and into your headwater streams, right? And so these events, these precipitation events, are really important for moving DOM off the landscape. And you know, I just wanted to stress like how important this hydrologic variability is in our world, in the world of streams. Like we have a really hard time like coming up with climatology of our systems because discharge is so variable, right? However, most of the times you go out to a stream, it looks like base flow. It hasn't rained in the last 12 hours, and the stream looks very similar on most days. But with respect to chemistry, these punctuated discharge events, right? This is what you're seeing here for this watershed in Connecticut for the last year, are become really important for understanding what's coming off the landscape because the concentration changes with these discharge events and because the volume of water is so high, obviously, when you have these discharge events. So in part, you know, this is what Jim and I started to key in on this paper is, you know, if you want to understand the movement of carbon off the landscape and you just go to your stream once a month and make a measurement, you're not going to get these dynamics, right? You have to focus in on these events. I had a master's student right around the same time, Brian Yoon, who set up an ISCO on the Esopus River. This is like this little R2D2 unit for like sampling the stream when you're not there. And it just so happened it was out there when Hurricane Irene went over and somehow wasn't completely destroyed. You can see what the stream looked like during Hurricane Irene. If you haven't heard of it, Esopus Creek is a sub-watershed of the Ashokan Reservoir, right? So supplies drinking water to New York City. And we found that even during that event, 500-year event or whatever, the DOC concentration went up and did not dilute. And we found that this particular storm over 2 days exported about 40% of your average annual DOC export. And so this was another eye-opener. It's like, not only is it common across systems, it seems to hold sometimes, at least, even for your largest storms. And then we had Henry Wilson right around the same time. He was a postdoc. This is what one of those ISCOs looks like, those little R2D units, on a stream in Harvard Forest. And he was really also interested in how the chemistry of the DOC changes with events, but he took great care to sample every single event over an entire year. And not just one sample, he would get samples over the entire hydrograph because we had this ISCO sampler. And so what he found was that this response with DOC going up with discharge held in the Harvard Forest for every event, but it wasn't the same all year round, right? If you have a similar discharge in March, right, you get an increase, but your concentration doesn't quite get as high as it would in October. And this is partly because in October, there's a bunch of leaf pack that just went down, right? And there's a greater amount of organic carbon to make tea with. And so this was also really interesting, though, because it was like a really sort of initial good first study that really went after a bunch of storms over a single year and demonstrated that this held. I'm going to skip this one. And so then, there were some conversations, and so, okay, we're like, this seems to be common, right? This idea of this pulsed nature of DOC off the landscape, what is there sort of in the conceptual literature that deals with this, particularly at the watershed scale again? For stream scientists, like, the first place you go look for this type of stuff is to the river continuum concept. This is sort of a real breakthrough paper that came out, I think it was 1980. And it dealt a lot with how river systems change over the drainage network, a lot more with ecology than biogeochemistry, but it did focus in on DOC. It did have a little bit where it discussed with DOC. And it stipulated that basically you have DOC coming in with headwaters, and it would be like slowly changed and removed, and chemical reactions would occur as you move down to larger in systems, so that its nature would change and it would be removed, and you'd see less and less of it as you move down in stream orders. I think which makes sense, but it really didn't talk about events, right? And we were like, well, you know, events are super critical for moving DOC off the landscape, and how can we conceptualize the importance of this event stuff at the drainage network scale? And so Jim and I and a colleague, Bill Sobczak, who's at Harvard Forest, we wrote a conceptual paper and came up with a conceptual model that I'm going to walk you through that we called the pulse shunt concept. And it's basically like, I think I've been over this. DOC export is dominated by events. But the other really critical part of this is, like, with an event, the velocity of water goes up. Right? So if you're standing by a stream and it's base flow, the water is moving slow. If you're standing there and a storm just happened, the water is moving fast, right? And so we wanted to layer this in. And so basically, we wanted to come up with a model that explored how these two things went together. Increasing flux and concentration with event, and increasing velocity of water and faster transport scales. Because the important thing is that if you're thinking about the reaction of a chemical within a system, there are really only two things that matter: the residence time that it's there, and the reactivity constant, or how fast it's reacting. And so this residence time was something we wanted to explore, and how that varied over the whole drainage network and with events of different sizes. And so we hypothesized within this that there was a shunt, right? And that not only is the most organic carbon coming out with these discharge events, but the velocity is so high that it's not spending enough time in the drainage network for removals to occur, so it's being shunted out to its receiving water. So, we built this model for this conceptual paper. So we built a conceptual watershed, basically, a representative New England watershed. And all the model had to do was route this DOC into the network and then follow it down, assigning travel times and velocities and assigning a decomposition constant as it went down. And so effectively, we were at first defining some of the scaling laws for a representative Connecticut watershed, right? And so I'm not going to go into these in details, but we were able to do this. And this is sort of a new thing that folks can do at the regional scale due to sort of these explosion in hydraulic and regional DEM products that are available to the community. There is another thing you have to do, which you can do with scaling, is you have to connect up a watershed. And you can do this with what's called the Tukanuga table, which also comes out of scaling laws of a self-similar system. And basically, all this table does is helps you determine how many first-order streams flow directly into a second order, a third order, a fourth order, a fifth order, and so on. And so this is dependent upon the geomorphology of your region, but it's something you can determine. And so you can start to see, right, if you're mathematically inclined, that these calculations that Jim and I done, you can do an Excel sheet, right, with these scaling laws that are around. And I think, we started off in an Excel sheet until I handed it to Jim, and then we got more sophisticated. And so, like I said, you have to flux the DOC in, and all you need for that are these relationships that I've been showing you. and information on the event statistics, which you can either easily gather up from USGS sites. So that's step one, the amount of carbon coming in. Oops. And then the amount of carbon coming out is just dependent upon the travel time, the residence time within the watershed, and the decomposition constant, right? There's different ways to express this mathematically. This is generally how oceanographers do it, so how I did it. And so this is called a first order exponential reaction rate, and this is your residence time. There hadn't been a lot of work at this time in around 2016 on drainage network residence time. It's actually a really tricky thing to do even still. But for conceptualization, we were able to simplify and do that by gathering up the average length of watersheds, of streams orders, and the average velocity within a stream order, right? So your residence time is length divided by velocity, how far you're going and how fast you're going. And we were able to do that. And basically, obviously, rivers get longer as the stream order goes up. Velocity also goes up, but the length scale goes up faster. So one of the key things that came out of this conceptual calculation is that the residence time in your first order systems was about 6 hours, right? So you're spending 6 hours with this sort of representative storm in your first order, where when you get to the seventh order, you're spending 2 days. And that is really something that sort of drives the conclusion, right? You just spend a lot more time as a water molecule or a DOC molecule in your bigger rivers than your smaller streams. And so this was sort of the last figure in the paper. Justin Farrell actually helped me make this, because he's better at making sort of network figures than me. And the way this figure works is each number and circle is a stream order. So, and the size of that circle is equal to the amount of DOC that's utilized in that stream order on a given year. And then the lines are the amount of that DOC that was utilized that came from the other stream orders. And there were a few things that fell out of this, but with respect to this figure, obviously, we're predicting that the largest DOC utilization is occurring in the bigger rivers, not the smaller streams. Not only that, it's only really the preceding 3 stream orders that matters with respect to delivering material to a given stream order. And this was somewhat important because there was this feeling that even at the 8th order, there was this community that felt like maybe first orders are really important at delivering fresh, new, labile DOC to the 8th order for consumption, but it's really only, if you're a 7th order stream, it's only the 6th, 5th, and 4th orders that matter for delivering material to you, just based on a stream volume. And this sort of law of stream, so that was sort of a law of stream subsidy that dropped out and had a standard number throughout the network. So I'm going to go over a few implications and then just talk about a little bit of data that's sort of been rolling in that we've made measurements to sort of see if this is happening out in the watersheds or not. So one of the big things here is that we presented a conceptualization that attempted to predict behavior at the whole drainage network scale. And this is something that's happening in my community. We're moving out of focusing on single streams and reaches or a single river system and trying to understand something at the network scale. And like I said, we're partly able to do this just because of how the spatial products have improved for us to be able to do this. And some of the instrumentation for making measurements allows us to do it at a finer scale also. One of the cool things for me was it was a great coupling of sort of hydrology, hydraulics, river hydraulics, and ecology, and with a real focus on events, in events and disturbance. And it placed an emphasis on travel time and residence time and how important this is. Like I said, this is a number that's still hard to calculate. Most hydrologists aren't that happy if you say, I calculated the residence time based on length and velocity, for reasons we can discuss later if you want. However, there are other studies, and I'll show some data, focusing in on trying now to match up travel time with field data to look at this sort of inversely. And some of the predictions we made, right, are that if you're interested in mass transport, you really have to be interested in events, and you have to sample events. And event statistics might be just as important as total volume of water coming out of watershed to determining transport process, right? And event statistics are changing due to climate change. We predicted that mass consumption occurs in rivers, not streams. Like I said, the stream subsidy. And we also had some predictions, which I'm not able to get into here, that looked at sort of the interplay between temperature and how temperature impacts that rate constant and events to determine uptake and export. Okay, so a little bit of data that's come out. We did receive funding to look into this in the Connecticut River. One really sort of great thing about the Connecticut River is it's kind of long and narrow. It terminates at only a sixth order because of this shape, but it has these fourth and fifth order watersheds stacked up on it, right, that you go over if you travel up 91, that are almost like replicates, right? It gets colder as you go up, the land use changes, but you have all these watersheds of very similar size that you can measure as you go up. With the USGS, we had a probe out at the Connecticut River at Thompsonville that measured DOC. Not only that, it sort of focused in on the DOC that comes from that terrestrial DOC, not the internally produced DOC. And we saw, this is Matt Schultz's work, a master's student, we saw that this relationship with discharge and the concentration of fluorescent dissolved organic matter, which is this terrestrial fraction, had similar dynamics to what Henry Wilson was seeing in the tiny little Harvard Forest watershed, right? It goes up with discharge. It goes up more in the summer than it does in the winter. This one's a little harder to step through. This is some work from Jake Hosen, who's a postdoc in the lab. So one of the really interesting things in the Connecticut River, though, is if you look at the total DOC concentration, which is here, there is no variability with discharge. And this is something that my field has been interested in for a while. We call this chemostasis, when a concentration doesn't vary with discharge. And we're seeing this for DOC, which is kind of counter to a lot of the things I was just saying. Well, one thing Jake was able to do is we probed the chemical composition of the DOC and sort of used a mixing model. And if you look at the amount of DOC that is allochthonous, right? This is that cruel word that you sometimes see in spelling bees that is means the DOC that's coming from the terrestrial system. Its concentration does go up with flow, just like we saw on the preceding slide. And because flow is going up, right, it has this really strong relationship between discharge and export. But interestingly, the autochthonous DOC, or the DOC that's produced within the watershed by the phytoplankton and submerged plants, goes down with discharge, right? And so this chemostasis is maintained from this source switching, where external DOC is important at high flow, internal DOC is important at low flow. And so this was, some of the first evidence, demonstrating one mechanism for why we have this chemostasis with some good field data. All right, and here was another paper by Jake where we again used sort of organic geochemistry to look at the flavor of that DOC or DOM. And this was another collaboration with the USGS where we had watersheds from all over the US. And basically, what he also estimated a travel time for all these different watersheds. And because we had tiny watersheds and big watersheds, dry watersheds and wet watersheds, we had travel time that spanned like 4 orders of magnitude, right, from like months to minutes. And he found what these graphs show here is that the DOC that has markers of going through reactions, you see much more of that character, the DOC, in the longer residence time watersheds than the shorter residence watersheds. So it sort of drove home this idea of the importance of travel time as it regulates the amount of time for reactions. It gets a little complicated because if you have reservoirs or lakes upstream, those obviously might produce a lot more residence time, and so we had to account for that. Anyway, some conclusions. This was one thing where, you know, if you have the luxury of working on something for 20 years, you could start to like see some of these patterns and try to conceptualize how something works as an entire system. I think with this paper and a couple other papers that came out around the same time dealing with other elements, this idea of the pulsing and the variability of concentrations in chemistry with storms and with events has really become an area of focus in my community. Working in teams allows for better science, right? There was a large group of collaborators, including hydrologists like Jim, that allow us to put this together. And I think it helped to have a little bit of a catchy name in the title of the paper. And so, yeah, I'll end there and take any questions. Sorry, we're getting into
a question over to the right
so quickly. Yeah.
And I'm wondering if, you know, that dynamism, and I can only imagine how hard it would be to measure the flux, especially after these pulse events, but the emissions of CO2 and methane from the rivers, you know, do you think it would be higher post-pulse events, or do you think it would get balanced out by, like, with the sort of net effect similar to between pulse events?
Right.
Because of that increased velocity and that increased effect?
Yeah. Yeah, I had a PhD student who focused on this for her PhD. So we have some knowledge. I mean, it has an added layer of complication because DOC doesn't have a gas, doesn't exchange with the atmosphere. And the other thing that happens is as velocity goes up, turbulence goes up, so the exchange rate gets faster. But in general, what we saw is that the concentration of methane dilutes with flow. And the concentration of CO2 either stays the same or goes up with flow. And so we interpreted that to be in part that methane is building up and being flushed, as you say, and evaded, where CO2, there might be a response within the system that's also adding CO2 during the event. It's a great question. Oh, sorry.
I just had a question about, you're looking at a lot of affected rivers, like historically that would have had more beavers and more, you know, woody material kind of slowing down flow. Do you think that has there been any comparison between, you know, rivers that are cut off from their floodplains and that are flowing more freely versus rivers that have been less impacted? And how do you think that kind of might affect the rate of DOC, especially like DOC that hasn't undergone reaction moving further downstream?
Yeah, that's another good question. So the model that Jim and I made in our representative 8th order watershed didn't have any connectivity to lakes or ponds or any of that. So it's not actually that representative, right? And I did do a follow-up student with a student at UMass, where we connected lakes, at least, to the system and small ponds. And we found that basically if you take CO2 and route it off the landscape, about 25% of that would be evaded within ponded systems and lakes and ponds. And so if you decrease the amount of ponding, you will sort of increase the travel distance of the gases. And it will be the same for DOM. We found that lakes are also, because a lot of the sediment drops out, they're much more reactive, at least photoreactive with respect to DOM. So it seems like they're very active sites for removal also of DOM.
Thanks. Yeah, I guess you were just touching on this, but I was curious about the relationship between DOC and sediment, and because sediment flux into streams can be really event-driven as well, but that might vary from one watershed to the next, depending on geology, land use history, agriculture. And I'm curious, looking at a meta-analysis, if you've seen, thought about seeing, effects from sediment availability and flux.
Yeah, another good question. So yeah, the concentration of sediment, or POC, usually goes up much faster than DOC, in fact. It has different memory effects, right? Because it's not produced quite as quickly as DOC. So if you have one storm and then a storm later, you might not get the same response. The bummer about sediment is it settles out and then resuspends. So modeling that at the drainage network scale is much harder. And so the dynamics are complicated by the fact that it's just not traveling around with the water itself. So that's partly why I haven't tackled it. But it does have this very event-driven component, and it's usually more event-driven than DOC.
Thanks. Two-part question for you. First, does DOC sort of serve as a proxy if you're interested in looking at pesticides or phosphorus or nitrogen or other things that are coming from agriculture, and to try and model those? Or is it sort of specific in how it reacts and you can't really get a sense? And the second part is your work being incorporated in other sort of physical carbon models that take into consideration not just rivers, but sort of the broader picture of carbon cycling?
So the first part, like for elements that are highly associated with DOM, it is used in the modeling. Mayodian, who's in the back, just did that global export of mercury export, and in part was trying to determine how much POC and DOC is being exported, and then assuming a concentration of the amount of mercury that's often attached. So it can become very useful. It also does have DON and DOP, and you can assume C to N and C to P ratios, and that's done. With respect to the modeling, there aren't a lot of drainage network models that I think are really doing a good job yet at event-based export. And so, yeah, I would say, and there aren't even a lot of models yet that are existing at the whole drainage network scale, but they're evolving. And right now they're just sort of like this, they're probing and coming up with different concepts based on those outputs. I mean, one of the issues is it's really, really hard to go out in the field and validate, because like our simple model, like you're assuming the Connecticut River gets an event of the same size over the whole watershed. That basically doesn't happen. So to actually watch an event evolve over an entire watershed like that, it just doesn't happen. So it's really, really hard to validate.
Thanks.
Hi, Professor P. May I ask, is there also a relationship between discharge and the flux of other inorganic forms of like carbon or nitrogen?
Inorganic?
Yeah, the inorganic part of carbon chemistry.
Yeah, so most of the inorganic and nitrogen, inorganic phosphorus, and alkalinity, inorganic carbon dilute. with events. There are representative relationships, but they're the opposite, where they dilute. All right, cool. Oh, go ahead, Paulo.
Yeah, great talk. I was wondering, we showed the relationship between this chart and DOC. It changes over the course of the year because of knowledge, because of processes. Yeah, so you showed the relationship between DOC and discharge and how it varies over the course of the year. And I was just wondering if there are some big changes happening across these broad watersheds that in changing that relationship as a background, not only during the major peaks, but just is there wood encroachment, changes in agricultural practices, that you see some transformations in that relationships, not only during the peaks, but the baseline over time?
Yeah, so DOC is changing. Rivers there's a term called brownification. It's kind of not the best term, but rivers are becoming browner, in part due to having more DOM in them now than they did before. And this is particularly around here in parts of Europe. And there's different hypotheses for why that is, but the leading one is that it's because of a decrease in sulfate in rainwater, which impacts the dissolution rate of the water once it hits the ground. I'm not aware of anyone who's shown an actual change over time in what we call the C to Q relationship, but they'd all be presumably shifting down, or I'm sorry, shifting up. But I haven't seen that. You need a lot of data to be able to show like a statistically significant change in your C to Q relationships. But that is the biggest change, is a response to cleaning up sulfate in the atmosphere.
I think we're gonna end there and give you another round of applause.